Hello everyone, thank you for coming to my presentation. If you're listening to this video, it means that I was not there to give the presentation in person. However, I wanted to share the highlights of information that I think would be most useful for people in the audience, especially for cultivators and those trying to make informed decisions about 2026 and beyond. The original presentation would have been 45 minutes or so, but this one is about 10 to 15 minutes. This is Cannabis Pest Strategy, Everswarm Evolution. My name is Matthew Gates. I've been doing IPM or Integrated Pest Management support for about 17 years now as Zenthanol Consulting. I've worked in cannabis, but I've also worked in gerbera, roses, lilies, tea, peppers, and a lot of other crops as well. I've written a lot about cannabis pests, currently heading a research report, a systematic review of neem use in cannabis and understanding the strengths and limitations therein. And if you want to contact me, you can find me at zenthanol.com. You can also find me on social media. I have a YouTube channel, Zenthanol, and I also have social media accounts on X and Instagram at SyncAngel. But you can check out the QR code for my website. When I talk about pests, I like to use this metaphor, the Everswarm. It is essentially acknowledging that all the various pests that we deal with are kind of a ever-changing, ever-evolving swarm of organisms. Not just insects, not just mites, not just fungi or bacteria, but all of them, the various subpopulations that exist, and this is very important, and how they move across the earth. So there's like a spatio-temporal aspect to it. There's time and space to consider, and what are our options within that context. The way that you grow might be different from the way another person grows or another group of people grows, but their effects can very quickly and rapidly ripple out and affect us. So we have to be cognizant of all of the different changes that are happening in the world, and as the world is as connected as it is, even more connected than it was when all the continents were together, to be honest, the movement of plants and the various pests that we consider to be pests are important to document. And in cannabis in particular, we mostly use research from other crops. So I want to highlight some of that research that exists now and some considerations for the future of research in cannabis. I want to say up front that the best thing that you can do is understand the pest capabilities and vulnerabilities that you're going to have to exploit. A robust IPM program is proactive and reactive. It has to have components for both because we can't just assume perfect conditions. That will usually come in the form of supplemental biocontrols or natural biocontrols that exist in your environment, various kinds of molecular or genetic priming using microbes or maybe certain compounds or proteins, and resistance breeding, both confirmed resistance and perhaps speculative resistance, since it's not always easy or possible or even tenable to check for these kinds of things or even have the resources to cross-reference. I also want to say that inflexible perspectives about pests conform our expectations. It amplifies confirmation bias. Ideas like optimal health, meaning that pests can't threaten your crop, or that using certain organisms like microbes or biostimulants or things like this make sure that the plant is enabled to be affected. Similarly, it's important not to go too far in the other direction. For example, the actual pest presence and threat profile for your region, for your particular property, for your facility, versus maybe a desired presence or threat, which is perhaps minimized or maximized. But you have to be aware of your situation. We also had to make room for potential contradictory evidence that would challenge our preconceived notions, but I would say that we should have more IPM research in general before we really tackle this too much, with a small caveat that perhaps the most initial research reports that are maybe funded by commercial interests or things like this should be interrogated a little bit more than surface level. A pest survey from 2023 found that fungi were the first level pest of all the different regions, the 5 regions that they split up here, as you can see. A lot of these are pathogens people have heard before, like botrytis and other bud rot pathogens. Powdery mildew, fusarium, and septoria is starting to become a real hassle. Also, there are oomycetes like Pythium and Phytophthora, which are also super common in other crops, and many people have definitely heard of them. But if you have not heard of them or you don't have any plans for mitigation, you should consider these because they are very common. As far as arthropods like insects and mites and things like this, This is a little bit split, but it seems like in all regions, moths are a pretty high contender for insects. Some of these are species that I've talked about at length, like the corn earworm, the European corn borer, the fall armyworm, the hemp borer, and the stalk borer. And some of these have close relatives in Europe or are already in Europe in different places. Hemiptera, or bugs like aphids and stink bugs and ligas and leafhoppers, were also found to be pretty common, and the Thysanotera, or thrips, were also kind of included in this. So 15% plus the 8% thrips equals 23% of all survey responses. This figure shows incidence of different pests in the 5 regions of the USA. A lot of these are pretty standard: bud rot, botrytis, powdery mildew, but it's concerning to see things like cercospora leaf spot or other kinds of leaf blight fungi become so prevalent, especially with a dearth of options available. Also, beet curlytop virus, hop latent viroid, and other viruses were found between 30 to 40%. This is also pretty staggering, and I would expect that, and I have definitely heard from growers, this will be increasing perhaps a lot more. A 2024 report for indoor pests in Florida found that hemp russet mite, silverleaf whitefly, impatiens thrips, two-spot spider mite, greenhouse whitefly, cotton aphid, cannabis aphid, and the infamous rice root aphid were pretty major. There are a few takeaways that I want to make clear. First up is the mention of impatiens thrips. It's possible that impatiens thrips is more common in certain regions of the US than others, and the same might be true for Europe. I know there's a flourishing floricultural zenith, especially in the Netherlands and several other parts of Europe, so this might be one to consider. If we look at the kinds of organisms that are being found in the survey, we see that just like the other survey, the Hemiptera, or the bugs, are pretty dominant. But interestingly, the moths were pretty dominant too. They were second in indoor pests. That's kind of odd to me. If we go down the list of highlights, the kinds of insects that you might consider to be more likely based on this research would be things like aphids, scale, whiteflies, moths, beetles, thrips, grasshoppers, and other locust-like insects, flies, wasps, and perhaps ants, earwigs, mites, and snails. The quick highlights are that fluid-feeding insects are on the rise, and this would likely account for an increased virus and pathogen transmission. Aphids are, if you didn't know, probably the number one plant virus vector of all insects. The closest might actually be certain whiteflies and scale insects, which are also Hemiptera, and thrips, which are a very close relative. Also, it seems like caterpillars such as armyworms are on the rise, and this would be a more environmental exposure threat. So if you have a greenhouse or if you have any kind of other exposure to these sorts of pests, it might be easier to get into your crop. That being said, for some reason it seemed like indoor crops were also being affected by maybe certain small moths getting in, so biosecurity is really important. Briefly, I want to show some examples of what these insects look like. Certainly I will make these slides available, and with the magic of video production, this can be paused and referenced for anyone who is interested. I'll also try to get this information up on my YouTube channel, so you can check that out, YouTube channel Zenthanol, for more of this information. But here we can see aphids, scales, and whitefly, moths, thrips, and certain mites. Here are some of the beetles they found associated with cannabis. Interesting to see a weevil depicted. I've definitely come across a lot of chrysomelids or leaf beetles, not just in cannabis but other crops. I know that a huge problem in several cruciferous crops, and particularly as larvae, they can be a big issue in cannabis. I find that in cannabis, the adult form is not necessarily a huge issue unless you have young plants in an outdoor setting. I do know that the Japanese beetle is becoming a major folivorous problem, and they're able to defoliate a lot of different crops. But if you were to ask me how often I encounter it, it's pretty low. A research report focusing on Germany looked at some of the new crops and pests that are associated with them, including hemp, quinoa, and ginger. One of the major takeaways is that an effective inspection system, robust quarantine procedures, cross-infection crop risk, and resistance testing would all be really important for these crops. And I agree. Briefly, I want to go over aphids, and this slide sort of encapsulates some of the important points about aphids. One is that they can be a vector. In this case, this is a cannabis aphid on the right that is vectoring or somehow moving russet mites on its body that it picked up. I've never seen this behavior in other aphids, and I've never seen this behavior since documenting this, but it's an interesting example. Usually aphids are vectors of pathogens, though, but maybe there's some way that these two specialist pests could collaborate. It wouldn't be the weirdest example of a cross-species symbiosis. On the left, we have lady beetle larvae, which, depending on age, species, and target aphid, can feed between 50 and 200 plus aphids per day. That being said, I'm not a huge advocate of using lady beetles, so I bring up this point because although this stat looks interesting, and I do know there's a lot of restrictions in their use in biocontrols generally in Europe, it might not be worth it for a lot of growers, and it's more of a reference of how the sort of wisdom of biocontrols can be very context dependent. But I have some kind of cool news, and that cool news is that aphids seem to be on the decline in a lot of places, at least in North America, and possibly, for example, in the UK. It seems like mostly pest species are actually on the decline, which is very interesting. We see in the Midwest and the Northwest this is pretty severe. But also, non-pest and keystone species are also affected, which may have ecological ramifications. Many of the locations that were sampled were down between 31 and 50%. You might be wondering, well, why would aphids be beneficial for me to keep around? There's ecological reasons, but one of them is that they are often alternative hosts for a lot of parasitoid wasps and other natural enemies that might be keeping the population more in check than it already was or wasn't, depending on your particular region. But if these wasps are given resources like honeydew from other aphids and water, then they do a lot better than if they're starved. So this is just an example of some of the ecological considerations we can use, even if we're mostly growing indoor and even if we're mostly growing in sort of insulated environments. One interesting note about plant resistance that I don't think gets a lot of attention is for cannabis aphids. Unfortunately, I feel like cannabis aphids they can be pretty easily dealt with, but it seems that CBDVA populations of cannabis, those with a lot of cannabidivirinic acid, seem to do a lot better at decreasing the survival of cannabis aphids. Different cannabinoid profiles and indeed different volatile organic compounds and similar substances, CBDVA might be an interesting research target for other kinds of pests, specifically insect resistance, but research like this shows that compounds that are already found in cannabis can have a strong effect on different pests, and they may even be beneficial for production. So that's a win-win. Budworms are a huge problem, specifically the corn earworm Helicoverpa zea and the cotton bollworm Helicoverpa armigera. If you've never seen them before, because honestly, usually people encounter them in outdoor environments, this is a large mature larvae. And then we have on the left the large chunky moth and the egg, which is quite small. We can also see depictions of different colorations of the same species, which makes them sometimes difficult to identify if you don't know what they look like or what they can look like. The bottom line up front, though, is that budworms have a cacophony of different resistances, both chemical resistance, Bt bacteria resistance, one of the only biocontrols that are really effective against this budworm, and also cold environment resistance that might make them much more suitable or habitable in different regions that otherwise they wouldn't have been. They're already hybridizing populations in Central America and the Great Plains of the USA, and I would expect as part of Eurasia, this is probably already the case in many parts of Europe, as it is certainly, especially with cotton bollworm, a huge, perhaps number one pest of other crops. They're so good at resistance that they even are able to deflect plant defenses and also substances that would have more general biocidal effects that are even targeted against other kinds of organisms like acaricides for mites. In Xinjiang, China, there are populations that are able to withstand minus 20 degrees Celsius conditions as pupae, and then when springtime comes, they come out as moths a lot earlier than it would have been for other populations to come out in warmer conditions and then travel great distances. They're one of the only pests I know that gets the descriptor megapest in other research, which I think really punctuates the point I'm making. The next members of the Cannabis Everswarm are the powdery mildews, specifically species in the genus Golovinomyces, but possibly others too. One interesting fact about powdery mildews is that they are closely related to another group of fungi called spider puffs, at least that's what I like to call them. The Arachnopezzaceae, if you translate their name, is roughly spider puff. I like spider puff. Basically, they are organisms that can break down plant materials and make them useful for plants. Usually, we would consider these beneficial organisms, but powdery mildews evolved from a common ancestor with these organisms. They have shared genes that allow them to both break down the cellulose in plant cells, but also communicate through the same pathways that plants rely on for things like mycorrhizae and other beneficial organisms. The problem is that this means that there are so-called susceptibility genes, and it means that a lot of plants actually have these S genes that are exploited by various pathogens. If they go away, resistance can occur, and often does occur. Very robust resistance, but it also makes the plant incapable of interacting with certain other beneficial microbes. So the effect of that is hard to predict. Powdery mildews have about 100 million years of temperate distribution, and they are really commonly found in Eurasia. And in particular, there are a lot of documented cases in China, in India, in Russia. But also, there are a lot of examples, at least sampled, in North America. Some of the other powdery mildews we might consider from other members of the Cannabaceae family includes those like the hackberry, like Celtis, or those of pigeonwood and trema. There are a few of those species, and we might consider researching to see how easy it would be or if they're even capable of colonizing cannabis. It's also important to note that at least a very ancient haplogroup of cannabis originates around eastern Eurasia and then perhaps moved across the rest of Eurasia and to other regions. So this sort of coevolution might mean that there are several powdery mildew species that are more apt to colonize cannabis in the Eurasian region. But you never find the same spore twice anyways, and they have a lot of time and sort of interseasonal development that plants don't get that allow powdery mildews to sort of go even further beyond their capabilities. They have these dynamic genomic compartments. They are groups of genes that they use, like tools in a toolbox, to manipulate the plant at a molecular level. That's very hard to deal with, and if we're looking for novel ways to deal with powdery mildew, we might consider exploiting this interaction in a more detailed way. But some of the species that we might consider to be an issue more exigently are the lettuce powdery mildew, Golovinomyces siceracerum. It has over 100 host species, and it's really good at hybridizing with others of its same genus and even other species of different genera. There's also the immortal powdery mildew. It has about 25 host species, but it's unclear about its taxonomy. It might have more, it might have less. And then we have the hop powdery mildew, which colonizes strawberry and caneberry, including hop. And there might be other species and other crops, for that matter, like hop, that might make integration with cannabis more difficult. As a quick example, this table shows the lettuce powdery mildew with a sweltering 61% transposable element content. That means that about 61% of the genes are part of this like highly mutational blend of exploitation genes that colonize and take control of the plant at a cellular level. You might have heard of some of the cannabis resistance genes that have been isolated or developed in certain cultivars. And I'm actually very excited about these. It should be noted, though, that in a lot of crops, if you're not aware, resistance can sometimes break under a number of conditions. Sometimes it's environmental stresses that make a resistance more limited. It's also very common for different populations of pests, and in this case, powdery mildew, to break the resistance with their own adaptations. This is about 100 years of melon powdery mildew development under different cultivars, and every few years, and sometimes almost the same year, powdery mildews can develop and have developed their adaptations to these resistances. Now, that being said, we shouldn't equivocate 1937 capabilities to 2026 capabilities of resistance breeding. But I think it paints an important picture that resistance has to be quantified so we can shore up those resistances better, pyramid those genes, and be informed when we're looking at resistance in different cultivars and deciding how much stock we want to put in their capability. Year to year, season to season, it could change. For example, these hemp cultivars were shown to have a spectrum of susceptibility. We have increased susceptibility on the right, white CBG, cherry wine, going down to Futura 83, Auto 2, Bayox, Futura 75, Fibra 79, FL 58 being way less susceptible in this example. That was 2023. In 2024, Photo CBD was found to be quite susceptible, but there were also examples in cherry wine, Carmanola, ACDC, Auto 2, and again, Fibor 79 being quite less susceptible. So we should be aware, even purportedly resistant populations, especially if they have been bred together or there hasn't been enough quantification, like I keep saying, we might not know exactly how it matches. And there's also other powdery mildew species out there that may be more or less capable than the one that was tested here. As an example of research that I wholeheartedly support, this is an example of a growth promoting bacteria called Peribacillus frigotolerans C1141. They found thousands of growth promoting genes, direct effect genes that do things like increase metabolite production. They degrade xenobiotics or foreign chemicals that might be detrimental to cannabis or to it, and also the solubilization of important nutrients like phosphate and potassium. They also had a lot of indirect gene effects. 200 plus genes associated with abiotic stress neutralization, 550 plus genes for plant derived compound usage. Those related to even its own fitness or spore production were found to be in excess of 100. So this is the kind of quantified microbiome research that I think we could do in Europe and abroad, and it can have some very important effects, very quantifiable effects. Compared to other plant growth promoting bacteria like Serratia plymoutica, Peribacillus frigotolerans increased biomass by 260%. It increased stalk diameter by 96%, and height by 43%. That's pretty impressive. A research report looking at different domestication grades of cannabis found that there was a core microbiota, Bacillus, Pelomonas, Ralstonia, Staphylococcus, and Pseudomonas. The first and the last three are pretty household names for bacteria genera. Many people have heard of species in these groups having positive effects, and also, especially in the case of Ralstonia, negative effects. But if you're looking to cross-reference different products and looking at certain microbes you might want to focus on or target, these may be some of them. So at the end, I want to close out and say that integrated pest management is more than products that you buy on a shelf. It's more than the integration of biocontrols or something like this. It's all about strategic information for implementation. This allows for increased flexibility and options in the face of constant evolution that is constantly being selected for. Even the best biosecurity or physical implementations can be supplemented by this information. There also may be new regulations in the future that allow for different kinds of products, and perhaps those that are an even improved version that are even less harmful, even less disruptive than those now. There also may be more strategic use of these options. Different regions and organizations in the US and Europe, for example, can come together and all decide to implement a combined strategy that makes the entire area a lot better for everyone involved. Not only the people producing, but the people who are having to live around that production, and those that are then going to be end recipient of whatever production exists. Whether that's in flour, whether that's in concentrate, or whether that's in oilseed or livestock feed or other sorts of uses. You have to use the landscape to your advantage. And although I made a great effort to make sure this information is both valid and relevant, there will be new information that comes out. So be aware of it. Verify all sources for claims. Not all information is equivocal, especially in cannabis. And just don't roll over for no data. Those things are really common in the cannabis space in my experiences, so if you don't do these things, I think you'll be doing pretty good. As Sun Tzu or Cao Cao or Du Yo or some other editors of The Art of War said, victorious warriors win first and then go to war. Defeated warriors go to war first and then seek to win. Have a good theory, have a good strategy, and you'll win before you win. As a modern philosopher once said, non cognosis si non inveris, non lucebis si non niteris. You'll never know if you don't go, you'll never shine if you don't glow. Thanks for listening, everyone.